All right, finally in 13.5 and 13.6, we're going to talk about antimicrobial mechanisms of action, so how the drugs actually work. We'll talk primarily about penicillin, and then in 13.6, we'll talk about how antibiotic resistance can occur. So here's a table of several targets of antibacterial agents. We can see things like cell wall synthesis, so making of the cell wall. That's something unique to bacteria, right? We don't have peptidoglycan. Cell membrane integrity. Polymyxins are in there. Those are in Neosporin. Mycolic acid synthesis, that's for Mycobacterium. Remember, Mycobacterium tuberculosis causes tuberculosis. DNA replication, transcription, protein synthesis, and different types of specific metabolism. So we see things in here you probably know. Penicillins inhibit cell wall synthesis. We've talked about rifampin and some of these other ones, trimethoprim in that multi-drug resistance case. So they all target things that are pretty unique to bacteria. These ones can be more difficult because they're very similar to what we do. We rely on the antibiotic killing the bacteria before it harms us. So if we were to look at this on a diagram, right, here's a cell. Antibiotics are going to target at least one of these processes. Usually they're very specific. So starting from the outside, right, peptidoglycan synthesis, cell wall inhibitors. Out here, things like vancomycin, penicillin. Cell membrane damagers, polymyxins like we talked about, they're going to hit here. Protein synthesis inhibitors that might target the different ribosomal subunits. Remember, bacterial ribosomes are different from our own, so we can target them. Things like chloramphenicol. Enzymes, metabolic inhibitors, and then DNA replication inhibitors, RNA polymerase inhibitors, inhibiting transcription. So we have these different processes that are key to life that we target because they are in some way different than what goes on in eukaryotes. So that's the selective toxicity, right? So in order for it to be selectively toxic, the enzyme or the process must be unique to bacteria. So DNA replication is not unique to bacteria, but the enzymes that do it are different from our own. So that's why we can target those processes. Let's talk about a specific example here. Here is the cell wall, right? Peptidoglycan, these cross-linked NAG and NAM with little proteins and stuff. So peptidoglycan is unique to bacteria. It's not in mammalian cells, so we can target this, right? And there's a group of antibiotics called the beta-lactam antibiotics that will target the construction of peptidoglycan. So there's an enzyme that builds these bonds called, this is a stupid name for it, but the penicillin binding protein is the name of the enzyme. It's named backwards, basically. Its job, the PBP's job, is to bind together the units of peptidoglycan to build that cell wall. So the enzyme penicillin, here it is one of these beta-lactam structures. They usually have some sort of ring in here. The beta-lactam antibiotics function to stop peptidoglycan synthesis. Penicillin, carbamazepine, I'm not sure what the other two are off the top of my head, but how they function is they bind to the enzyme and block its function. So this penicillin binding protein, it was named because we discovered that penicillin would attach to it. This penicillin binding protein, remember, it normally builds the peptidoglycan. So you could think of it as the peptidoglycan building protein. Penicillin goes in there and blocks up the ability of this protein to bind the units of peptidoglycan together. So over time, the bacteria can't make its cell wall, and the cell wall falls apart, and eventually the cell will burst due to osmotic pressure. So that's how penicillins or beta-lactams function to attack the cell wall and destroy bacteria. Penicillins work best on gram-positive bacteria because gram-positives make a lot of peptidoglycan, and gram-negatives have that second outer membrane that blocks the ability of the penicillin to get into the cell. So gram-positives are affected most by penicillin. So obviously, if there's an antibiotic, cells can evolve resistance to this. How can cells evolve resistance? We're going to talk about the specific mechanisms. There are two broad ways, right? They could acquire resistance genes through horizontal gene transfer, so they could get resistance genes from other organisms, or there could be mutations that occur in their own genome and then natural selection occurs. One gene that is commonly passed between bacteria is the gene for an enzyme called beta-lactamase. So penicillin is a beta-lactam. Beta-lactamase is an enzyme that degrades beta-lactams. So beta-lactamase chews up penicillin. So bacteria that have this enzyme, they encounter penicillin and they chomp it up and break it down into something that is no longer toxic to them. That is one way they could be resistant. The other way is for this penicillin binding protein. If it had mutations in it that allowed it to still function but prevented the penicillin from entering it, then it would now be resistant to penicillin. So random mutations in the gene for the penicillin binding protein, they could prevent penicillin from getting in there and halting peptidoglycan synthesis. So those are two ways that resistance could evolve. So if we look at it right, normally the penicillin binding protein is going along, cross-linking these peptidoglycan units, building the cell wall. Antibiotics, the beta-lactams, get in and block that up. If the bacteria has the enzyme, it has the gene to code for the enzyme for beta-lactamase, it will be resistant because before the penicillin can get in, it gets chopped up by this enzyme and broken down. I would like you to know this example for the test. Please be able to explain how this is functioning. All right, before we go on to 13.6, let's do a review question. Hypothesize why there are more chemotherapeutic agents that block bacterial protein synthesis than chemotherapeutic agents that block viral protein synthesis. So, hint, hint, think about the differences between bacteria and viruses. Is it A, viral protein synthesis occurs inside the host cell while bacteria protein synthesis does not? Is it B, viral protein synthesis relies entirely on host ribosomes? Is it C, bacterial protein synthesis relies entirely on host ribosomes? Is it D, viral protein synthesis does not involve ribosomes? All right, go ahead and pause the video. Think about this one. Remember that hint? Okay, so my hint. Viruses are non-living, right? They can't do their own protein synthesis. They need to hijack the machinery of the host to make proteins. So the answer would be B. Viral protein synthesis relies entirely on host ribosomes. That means we can't really target that process without harming the host at the same time. Bacteria, on the other hand, have their own ribosomes. They do their own protein synthesis, so we can target those ribosomes and not hit our own in the process. Okay, in 13.6, we're going to finish up by talking broadly about how antibiotic resistance can happen. We talked about a very specific mechanism there. We're going to talk broader about the general ways this can happen and how that can lead to drug resistance developing. So, I would love you to know these. These are the three basic ways that microbes can be resistant to antibiotics. And I'll show you an image in a moment, but I'm just going to go through them. You can prevent the intracellular accumulation of the antibiotic. Essentially, don't let it get in or keep it from staying in. You can prevent the antibiotic from binding to its intracellular target. Or, finally, if it does bind, you dislodge the antibiotic from its target. These are the three ways that bacteria can evolve resistance to antibiotics. So what does this mean? All right, here's our cell. We have various things going on. We have like a ribosome here. It's translating this mRNA. So the first one, mechanisms that prevent intracellular accumulation. We have a couple of different options. So this is preventing the antibiotic from getting into the cell. You could destroy the antibiotic. So this beta-lactamase, right? Remember beta-lactamase chewed up penicillin, the beta-lactam antibiotic. If the cell excretes that into its environment, it will destroy the penicillin before it ever gets near the cell. Another option is to pump the antibiotic out of the cell. So the antibiotic gets in, but then it pumps it right back out before it can accumulate in the cell. Or you can decrease membrane permeability. So you block the pores that would allow the antibiotic to come in. So those are all strategies to prevent entry. Our second option is to prevent it from binding to its target. So in this case, we have an antibiotic that binds to the ribosome. So you could alter the target, so you have a mutation that now no longer allows the antibiotic to bind to the ribosome. Or you could have an enzyme that kind of changes the antibiotic so that it no longer can bind to the ribosome. Those would be two strategies of preventing binding. Or you could have the third option, dislodge the antibiotic. So we have an antibiotic here that has bound to the ribosome, but a special protein comes in and like pushes it off and removes it from the ribosome. These are the three basic ways, and some sub-variations of them, that microbes evolve resistance to antibiotics. Obviously, this takes both horizontal gene transfer and random mutations occurring. And this is a problem. Antibiotic resistance is increasing. Although luckily, now that we're aware of it, we're taking steps to avoid this with things like proper prescription of antibiotics. So here's a graph that shows time down here and the percentage of clinical isolates that show resistance or intermediate resistance to penicillin. So in blue, we have penicillin-resistant bacteria. In orange, we have intermediate resistance. So there's always kind of been a background level of intermediate resistance, but it really started to increase in the early 90s. And then we also started to see an increase in fully penicillin-resistant strains, which actually peaked in the early 2000s, with about 30% of isolates showing penicillin resistance. There's many factors that go into this. Overprescription of antibiotics, improper prescription of antibiotics, people not taking full courses of antibiotics. Luckily, through identifying this, we have been able to reduce the number of clinical isolates. But you will see that it is still much higher than it used to be. Somewhere around 15% of clinical isolates are penicillin resistant. And that is evolution occurring. Once it happens, it's not going to go away. It's being selected for. So some of this drop would also probably be due to hospitals no longer prescribing penicillin, right? They find that 30% of the time it doesn't even work, so they stop prescribing it. So there's no longer that selective pressure there, and the strains might lose that resistance. In many cases, antibiotic resistance is only useful when the antibiotic is around. It might in other ways compromise the bacteria, so it's not the optimum state generally. So hopefully this will continue to drop, but at some level it's always going to stay there because antibiotics are a selective pressure. We are applying that selective pressure, and resistance will evolve. Remember the example we talked about in broad details. Here's an example of a patient who is sick with a bacterial infection, right? So they get some sort of pathogen in there, and it grows to a high number. They are now sick with pneumonia. Some sort of spontaneous mutation might occur, and now one of these cells or one group is resistant to an antibiotic. We apply the antibiotic, and the patient recovers, but these resistant bacteria start to grow and multiply, and the patient will relapse back into disease. This is because selection has occurred. Usually this relies on the immune system coming in and clearing these out, but in elderly people or people with weakened immune system, that does not always happen. So trying to prevent infections from even occurring is a great practice, right? Hand washing, vaccines, boosting our immune system. These are all great ways to help prevent drug resistance from developing. Okay, I want to finish by talking about antivirals. Antivirals, remember, work on viruses, as we talked about in the virus chapter. Many of them are DNA and RNA synthesis inhibitors. I want to stress that antibiotics do not work on viruses. So many people go into the doctor's office with viral infections, things like the common cold and stuff like that, and they want doctors to give them antibiotics. They want magic pills that will make them better. Usually the best course is if you're not deathly ill, you probably don't need antibiotics. Your immune system will deal with it. We don't have a lot of antiviral agents because they tend to function to disrupt DNA and RNA synthesis, both critical processes that are using our cells to do this. So these often have very strong side effects on the patient because of that. All right, that is it for our unit here. I hope you learned something about microbial diversity and how we try to control that microbial diversity in this section. All right, bye for now.